Hi, and welcome back to Bio24, where we're going to be talking about neuroscience. Neuroscience is going to pertain to a lot of future lectures, so this is going to provide a really nice foundation for a lot of what we're going to learn in the next week. In this lecture, we're going to be first talking about the nervous system, and then how it sends signals, and then talking about a little bit of issues that we've seen with the nervous system in the past. The nervous system includes the brain, spinal cord, and associated tissues. The brain is vitally important and makes up roughly 15% of the body's cardiac output, using up to 20% of the oxygen consumption, while being comprised of only 2% of the body's total weight. The nervous system is made of interconnecting neurons and glial cells. So there are two cell types that we find in the nervous system. The neurons, which you may have heard of before, and what nervous system is named after, are the primary cells, and they process and transmit information by electrical or chemical signaling. They are what really comprise a lot of the brain, spinal cord, and peripheral nerves. Then there are the glial cells. The glial cells are cells that support the neurons. They provide nutrition and also help conduct the signals between the neurons, making the signal conduction more efficient. The neurons are, of course, the primary cells of the nervous system. And so we're going to take some time talking a little bit about the cell anatomy. There are three major components of a neuron. There's the body of the cell, otherwise referred to as the soma. This is where the nucleus is located, along where a lot of the organelles are also found. The extensions from the soma include two different types. There are the dendrites, which take information from other cells and bring it to the cell. And then there's the axon, which sends information away from the cell. The axon ends up terminating in what's referred to as the axon terminal bouton. It's spelled like button, but it was named after a French discoverer, so we refer to it as bouton. The axon terminal is where the signal is being sent from the neuron to other cells. And we're going to be spending a lot of time talking about that. This is a classic neuron where there is one long axon extending from the soma and many dendrites bringing signals in. However, there are two major types of neurons based on their overall shape. Type 1 have this characteristic long, thin axon. These are the type that are found in the spinal cord. In the spinal cord, the axon is surrounded by glial cells. In this case, the glial cells are wrapped around the axon and form a structure called the myelin sheath. In this particular image, there are 5 separate glial cells in purple that are wrapped around the neuron. You can even see the nuclei forming a little bump on each one of these. This myelin sheath comprised of glial cells helps the signal transmit along the spinal cord faster and more efficiently than if they weren't there. It's like the equivalent of the covering to an electrical wire. Type 2 neurons have no long axons. They are the gray matter of the central nervous system and send and receive signals in short distances, so from one cell to another in your brain. Neurons can also be classified based on how they function. There are 3 different categories, the first of which that you see here is the motor neuron. Motor neurons receive signals from the brain and spinal cord and send those signals to tissues of the body, sometimes causing muscle contractions, or in other cases, altering gland activity, sending and receiving hormones. Then there's the sensory neurons. Sensory neurons receive signals from the body and send them to the brain. They respond to touch, sound, light, and other stimuli. So we have signals being sent from the brain in the motor neurons, signals being sent to the brain, that's the sensory neurons, and then of course we have the neurons found within the brain, and those are your interneurons. Interneurons connect neurons in your brain to other neurons in your brain and also within the spinal cord. Neurons transmit signals from cell to cell. These signals can be in the form of chemicals, such as molecules, or an electrical current. If they are sent as chemicals, these chemicals or molecules are referred to as neurotransmitters. If the signal is sent as an electrical current, that electrical signal is comprised of ions or charged atoms, so much smaller than the neurotransmitters. Because neurons transmit signals, either neurotransmitters or electrical ions, from cell to cell, they must move these molecules or atoms across their cell membrane. They do this by using protein transporters, proteins embedded in the cell membrane that allow molecules to flow across. So there are two ways to send a signal from one cell to the next. The first that we're going to talk about is neurotransmitters or chemical signaling. The signaling occurs between cells like neurons. This can also be between a single neuron and a muscle cell or a cell in a gland. The junctions between neurons is what's referred to as a synapse. So a synapse is the location where a signal is sent from one neuron to another. Neurotransmitter molecules are stored in small bubbles called vesicles in the initial cell. You can see these here, where the molecules are located in these little membrane bubbles. When the first cell is signaled to release the neurotransmitter into the synapse, the neurotransmitter enclosed in these vesicles will move toward the axon terminal button and release the neurotransmitters into the synapse. The nearby neuron receives the neurotransmitters using protein receptors located on the surface of the cell. As you can see here, the signal is being sent from the axon in the first cell to the dendrite in the second cell. The neurotransmitter must then be eliminated from the synapse in order to have the signal reset. There are two options to eliminate the neurotransmitter from the synapse. One is pictured here, where there are enzymes that outright destroy the neurotransmitter. Another alternative is that the first cell that sent the signal can reabsorb the neurotransmitters into its axon and save them for use again later. A common neurotransmitter is acetylcholine. Acetylcholine goes by the abbreviation ACH. It's used by both the central nervous system and the peripheral nervous system. The central nervous system, or the CNS, is your brain, and the peripheral nervous system, or PNS, is the neurons that send signals to all the areas of the body. In the peripheral nervous system, acetylcholine activates muscle cells and plays a major role in the autonomic nervous system. It ends up being an activator of different tissues in your body. In the central nervous system, acetylcholine tends to cause excitatory actions, promoting signaling and long-term memory formation, resulting in permanent connections between neurons in the brain. Remember that signals can move along and between neurons by electrical signals as well. How does the neuron generate an electrical current? This, if you remember, is a result of a change in ion concentration. The most common ions used for this purpose are sodium, chloride, and potassium. Sodium chloride is your typical table salt, and potassium is an ingredient commonly found in many foods, but particularly in bananas. Unlike electrical signaling, where specific signals are sent depending on a specific chemical that's used, like acetylcholine, dopamine, norepinephrine, and others, electrical signals only act as an on or off switch. So there is no specific signal being sent with an electrical signal. We're going to talk more about these specific signals and specific neurotransmitters later. These electrical signals are sent using what's referred to as an action potential. Neuronal cells, as you can see from this graph here, have a resting, stable action potential of minus 70 millivolts. This means that they are most stable when they have a negative charge inside the cell. That negative charge is a result of chloride ions, which are negatively charged, being inside the cell, and sodium and potassium ions, both of which are positively charged, being outside the cell. Action potentials are triggered when the positive ions, specifically the sodium ions, rush into the cell. That causes the cell to become slightly positively charged, what's known as depolarization. Then, to reset the system for the next signal, potassium ions rush out of the cell, causing the cell to become negative again. This is what's referred to as repolarization. As you can see, there's a temporary refractory period where the cell becomes even more negatively charged, making it difficult to generate an action potential immediately following one that's already formed. This keeps the cells from continuously being engaged with a constant electrical signal being sent. How can we determine how the brain works? We're going to talk about some studies in the next lecture about neuroscience from the early days until now.